So good morning all, uh, and hope that you can hear me. Can you please comment in the chat box so that way I can come to know that the voice is clear. Um, Today's session is very important as we are going to see the demo of the tools which you will be using in your project. So I will take one example uh, from the project and I will run the tool. You will be utilizing the tool, but one major thing which we have discussed in this week is that you need to make sure that um, your role is as a professional expert in this. So you will be not just going to take content from the large language models and not submitting. You are actually going to review it so that way it will confirm that the tools, the large language models are giving the content which is relevant to the topic. That is one thing. The second thing is that I want to show here that how actually the journey is moving from generative to agentic in Swalife Biotech, where we are developing product, and there where the purpose is that with the help of this information, you will actually learn the skills in AI, but simultaneously collaborate or create your own research group and replicate the similar type of work which we are doing, so that way it can be useful for the drug discovery area and for the general public. So initially, before starting with the demo of the predictive intelligence, I will show you a couple of our previous tools which actually help us to develop the next version, which I have shared the link to you in the email. Um, and from there, we actually now move towards the agentic. I have already shown you the agentic AIs, so we have moved further, but initially when we started, the purpose was to actually have a prompt which can connect the project. So I will show you first two tools which were developed, and then we had some user on it. We use them, and then we understand. Based on that, we move towards the second version, which I have given to you for the as part of this project-based program. So I will just share the entire screen. So when we actually moving from one platform to another, you can see. So this is the first tool which we have some preprint, and also some articles have also used this where it helps to generate scientific prompt to understand the target, lead identification, lead optimization, and preclinical study. We have not stopped using this. We are still using it because once we move into, so we initially start with the plant. We do the network pharmacology. We identify the targets and the lead. Then we move towards the this tool, and I will show you the next one. And based on that, I start with the predictive intelligence. So the teamwork in this flow, I have directly given you the predictive intelligence here because the molecules are already been identified and everything is already tested. And now you will be doing your further work in there. So here is the target identification. Based on network pharmacology, you will have one target. So let's say in this case, we will take PRP1, which is for DNA damage response pathway, and the prompts which will be generated will be 20. You can increase the prompt, and you can actually have as much prompt you want. but you have kept it minimum 20. So you can generate for identification, generate for validation, or you can generate for both. So once you click on generate for both, the interface is actually having some issues. Prompts have been generated now, you can see here. So I will just download it, and we can see that these are the prompt. So you can see the profile PRP centric mechanism within cell cycle control using. So this is the first prompt which is useful in target identification, and all these are for the target identification purpose till here, which will be 20 prompts, and then below that there are validation prompts. So this is how the prompts have been created. You can see that I only put the PRP one, that is one of the protein, and it has generated or it has created the prompt which are connecting to the identification process. So in this profile, quantify, integrate, so they are connecting to each other, which will be then moved with the large language model. This one design where it comes from target and it moves towards clinical and PV as well. So this was the one which actually covers everything. Now we actually got separate. I've given predictive intelligence for the basic discovery. Then there is a clinical studio for AI-based clinical studies, and then there is a AI-based pharmacovigilance. So we separated that, but previously it was in the one interface, but then the content wise, the one which given to you is more specific, and it actually gives you more relevant data compared to the one which we have got here, and that's why we updated the tool. Again, here you are going to put the disease and the name of the plant, chemical constituent, or herbal source. Here it only generate the 40 prompts, and you can see when it clicked on there, you will have this prompt one. So this is prompt one where it will search the databases to understand signaling, inflammation, onco, vulnerable pathways. Now this was more specific towards uh, the oral cancer. If you change the disease, let's say we will say diabetes. So if you click on generate diabetes for this one, it is again going to be more specific towards the curcumin, uracil, and ECG, or in the second one it is saying they build a disease gene list for diabetes. So it is connecting to the molecules and the disease. But what was the issue here is that the prompts which were there, it was more about some of the specific pathways, and that's why we wanted to make sure that when you are searching for certain other diseases than cancer, because this was more relevant to what we are doing. But when we want to actually, other people to collaborate and do things, we want to also make sure that different pathways are also targeted. We are not actually ignoring them, and that's why we are updated towards the new version, and that is the predictive intelligent tool, which actually covers everything and also help in predictive analyticals and predictive medicine. Now, how this work, I will show you with the example, and you are going to do a similar thing. But whatever you do, you should make sure. And one thing we always mention there is that developing the trust and making sure whatever you are having in the data is being recorded, noted down. So in this case, so let's say for example, I've given you the title for these three molecules and oral cancer prevention and oral gel. Okay, so when you are doing entering your data, we want that you do global in this case. But if you want to, have certain country like India only, you can do it. But you need to make sure that you are going to report that in your report, and also when you are creating your, the draft of your article. Then the population will be there. study types, there are different study types, but in this case we will be using literature review. And because I am doing here demo, so I have written down demo. Here your project name will be. Okay, so this is optional, but you should still enter here because it will be reflect on your tool. There is also option for notes. So like I told you that we don't directly come on this tool. We actually utilize this tool first and then move here. So anything we have seen in this previous tool, we actually put the note here. So that helps the tool to understand and design the prompt as that way. Now once you enter all this information, you need to make sure that you save it. So once it is saved, because I previously entered something, so there is already a prompt here. But normally there will be no prompt. Let's first actually delete this. So now there is no prompt. So once you save it, there will be still you can see there is no prompt here now. And when you once you save it, you need to go to now the prompt template, prompt studio template, where you need to select the first level, that is the complete evidence pack from biology to regulatory, and then you need to load this. So once you load this, you will see now the prompt has been generated. So in this, you can see that it is telling the large language model that you are the pharma R&D scientific analyst for Swalife AI. Build a complete structural evidence pack for the molecules which you have given, the disease, the formulation, the reason of the regulatory area, and the target population. Now the rules are pre-designed, so it will you know use the previous content, and based on that and based on this rule, it will deliver the information which is needed for our project. So now I have created the prompt. I will copy the prompt, and then there are the large language models which you need to click. In this case, we will be using Perplexity. If you have any question while the demo is going on, large language model. So let's say you're working on ChatGPT, Gemini, create projects there. Also on the cloud, there is a projects. Perplexity, there are spaces. You always create the spaces of your project. For the demo, I'm not creating, but for your project, you should create a space with the name of the project. Because what happened then, you whenever you have the prompts, you can put there, and it actually learn from those prompts. So now I will paste the prompt which was generated there. Now once you added the prompt, you should always in your project you should always go for deep research. For the demo, I am not going to go for deep research because it will give the information faster. But again, with the normal prompt search also, the information that you are getting is a part of the RAG AI, that is the retrieval augmenting. That means it is giving the information and also the citation of the articles from where the information has been identified. But still, when you are using the content, you should it has given us the reference. It has referred and given nearly 6 references here. Same thing, disease overview is given, which is again having citations. Um, Key clinical endpoint. Now, this information, why Perplexity or why LLMs are giving? Because we have the prompt designed for that information. So you can see that in the first, it has to give the disease overview, right? And that's why we are getting disease overview. It's not that the LLM has generated that information. Yes, it has, but we have asked that, and that's why we are getting that information. So you are going to get total 12 types of information, including the references nine, 10. The references are from the genuine sources and the references are not generated. So that is the role of our expertise here. So you can see about 30 sources it has been cited, and you need to make sure, things like this. It is a blog website. You can refer it, but you shouldn't actually include in the scientific documentation. And why? Because those are the blogs which can be deleted by the person, the owner. Simultaneously, those are the view. If they are not having the references cited and no references given, those will be the view from that person. So if you cite any blog which actually have the references, you can refer them, you can use the reference, and based on that you can put in your documentation. But blog shouldn't be cited. So you can see maximum references are from from the databases. About the blog, I told you one more. Now again, because this is a government from the website or a charitable website, you can cite them, because with the government, with the charitable website, they put the things based on the references and based on what they are working on. So definitely, those can be utilized. Now, this is the first step. You are doing all these things. Now, in the second step, uh, you need to copy this and you go back to uh, the tool, and you need to see here is the option there, paste LLM output. It says rating, but you don't have to rate. You just need to paste your complete content where you can remove the final version, which is given by the perplexity. You need to remove that. Okay. To improve the things, you can put what is missing, as I told you, because you are reviewing on perplexity and everything content. And then once you are done, you can see here there is option for save run. You can see here in the chain builder there is no save run. Okay, so when you click here there is no save run, but when you click on save run, now you can see that June 6 2026 at 11:18 a.m. there is a content from our first which has been saved. Okay, so you are going to save it. Now you need to remember this, and when running the tool, you need to always follow this, where you will have a table and the dashboard, and also the JSON file created. That is a part of a data analysis. You are going to do, you are going to go for level 2, and you are going to, before loading, I will show you again the first prompt, which is you are pharma R&D scientific analyst for Swalife Biotech. When you click on the load, it is going to change now, but not yet. Here you can see you are a scientific data curator now. So you are not analyst, you are now data curator. Convert the following literature mining output into strictly structural data. Now remember this. This is still not the complete yet. I'm showing you the the one stage early, and then there is a final one which is very important stage. Once your prompt heading is designed, it is going to give you the context which is in the main window. The input, see the input, it says input level 1 prompt, but it is not there. For that you need to click on here, build structuring prompt. So once you click on here, now you can see the input level 1 has been picked from the previous prompt, that is the complete prompt, and also the answer which the prompt has given. So that is the information which we have got from the any large language models that you have created. And now you got the prompt in the format which we want for the data curator. So you copy that, and then you move towards the space which you have created in perplexity, and you paste here. Again you will go for deep research, and you will submit. To convert this into a structural data. So you can see it has given the executive summary first, then it is giving us the table, the compound table for the compound which we have given, uh, different evidences, keynote, then the target table, okay, and then the safety table, regulatory claims table, and the JSON file, which is sometime needed, not sometime actually always needed if you are also running the same for any other LLMs. So that's your first content from your the first um, phase of the literature mining, which has given us more specific, more relevant information for the project which we are in. Once you got that, you are going to copy that content, and you are going to put into the Word file, which is your report. But while doing that, you are not going to just copy and paste. You are, as I told you, because the content which you got from now from Perplexity has to connect with the sources, and the information which has been given has to critically analyze by you while understanding that does these molecules are really having any effect or role in the disease which will be there from the article. Now this is the first stage for the predictive intelligent project. Once you complete the first one, you move towards the second one, that is the AI discovery and study designing. You are going to you are not changing anything here. It will be say already saved, it is already there. You're just going to start with target and lead hypothesis. Here there are three prompts. So two are for the for the data, which will be for target, leads, and study designing, which contains preclinical, clinical, PV marketing for the the chemical constituent and the formulation which we are developing. And the third one will be for putting in the the structural format. Now the structural format is our content which we be looking from the data. But you need to remember that when you are saving here. So every time when you run the studio, the second time, the first one will be saved as a input here. Okay, so remember this, there will be one input from here, and there will be two input from here, which you need in the third stage, that is in the predictive analyticals, that you will need the literature run and from the discovery run. So in the discovery run, you will have the content which will be saved here, and then you are going to develop the build analytical prompt which will be based on these two content. So why this was created? So that's why I showed you the previous two tools which were creating the prompts. But those prompts, when you run in the large language model, the critical question was what information I got, which I can generally put, but I wanted to put them into a structural format. And for each project, it should be the same prompt, and that's why we created this. But not only stopping here, also based on the data which you got from the literature mining, from the in vitro, in vivo, and clinical, also PV and IPR study of that molecule, because we are working on small molecules from plant. We will be developing novelty after that. We have got tool for that for others, but for small molecules from plant, our aim is to target multiple protein with multiple molecules, and there will be a formulations of numbers of molecules. So to understand that does it actually they have the target, we have this predictive analytical which connects with the literature and with other information about the in vitro, in vivo, and also other studies done on those formulation molecule, and it analyze and give us information that how much it is useful with that disease. Now we don't stop here. We move to the fourth one, which actually help us to design the personalized, predictive medicine. That is the decision making for the clinician, for the doctor, where it will decide on the information. So here you don't have any other, content which you need to run as a target, but you will be taking the prompt which you have run in predictive analytical. So the same will be here, and from based on that you will be building the prompt from that one. So you are connecting them each other, and then you will be having a final predictive medicine information which will be useful for the clinician, for the doctors, to make sure that can I give this formulation while when we are treating a disease in oral cancer for this compound. Why? Because let's say for example a clinician or doctor is treating a oral cancer patient with a allopathic drug, let's say 5-fluorouracil. While doing that, can I give these molecules? That is what we are going to get out of this tool and from this project. So those people who are doing project in productive intelligence need to come to a conclusion that what allopathic treatment and how these three molecules can be useful in oral cancer prevention. Okay, so that's about the predictive intelligence. So if you have any question, um, you can ask in the comment box. This is for the project which has been allocated to you yesterday. Okay, so that's for the AI in predictive medicine using predictive intelligence, where we are focusing oral cancer chemo prevention. When the actual chemotherapy is going on, patients are suffering from many of the side effects, and we are actually touching the base with the predictive medicine. But simultaneously with this information, we move into in vivo and clinical study. But we also have a predictive tool which can be then be useful by for the doctors to decide on can we actually use this formulation for the patient who has been treated with the chemotherapy drug. Okay, so now uh, we will go for the next tool. If you have any question, you can put in comment box. In the meantime, I start the next tool. Okay, so any question? Let's see. Okay, so there are two questions. Okay, so can you share me your name because we have allocated the project. This is part of the project-based program. If you are part of that, then you are doing project. But if you are collaborator or if you are someone who want to design your own original prompt, then you need to talk with us. It is possible to design the prompt. Okay, so you are part of the project-based program. So you need to actually utilize the project which we have given to you, and you will be. So which project you are doing? Okay, Dhirendra Kumar Singh, you are from the predictive medicine. Yeah. So I showed the demo for curcumin, uracil acid, and EGCG. Your three molecules you are going to add into the the. Let me show you. Actually, that is what I showed. This is the way you are going to put your create your own prompts. Okay, so here your molecules will go. Oral cancer chemo prevention oral gel is the formulation that we are developing, and then your prompts will be get designed. So that's the demo which I showed you. Only thing is that your molecules are different. That's it. As part of this project, if you are asking, for future or different area, then you need to contact us. You got the email ID, you got the team mobile number. You can contact us on that. Yes, you are using already design. Already design means for those plant and for those disease it will design. If you change the plants, if you change the disease, it will design for that. Right, that is how it works. The purpose is that you actually have the structural prompt, so you will get the structural information. If you change the prompting, you get different information, and that's what we are trying to reduce uh, when we are actually working on the formulation for certain category of the formulation. So, if let's say, for example, if I am working on oral chemo prevention or oral cancer, or let's say diabetes 2, in that case, I will design the tool for diabetes 2, right? But what we got there can be utilized for diabetes 2 as well. But now, if you want something which is not in the prompt, that is that is possible. You can directly ask the LLMs about that. So you will still get the information. What we have done, we have not we have moved further. We got the agentic now, but with this, you can always because the prompt which you are running on the LLM, you can continue the same, further questioning, and that is what is happening with the people that based on the information they get, you again go for next question, and instead of utilizing that data for the the study, you actually continuously ask question and you lost in that process. And we have in the previous we have tried this, we had like 20, 40 prompts, and it has created a mess, and that's why we have moved to specific. And now with the rag and with the agentic we have gone further specific. Okay. Okay, so that is about the um, predictive. Now the second one is the AI in clinical research. So let me. Okay, so this is the uh, Prompt Studios for um, clinical projects. So you will have this interface when you open your Prompt Studios. Your project is again for oral chemo prevention, oral cancer chemo prevention. You will be doing curcumin oral gel, and all the groups have been already allocated the name, but you will be working on the cross sectional observational studies. So what actually it is one of the. Let me just. Can you hear me? Hello. Okay. So what is happening here with the AI in clinical research? The interface which is given to you, that is the web tool for AI clinical prompts, and we have got different studies. But for this batch and for the project allocated, we will be focusing on the cross-sectional observational studies. Why? Because it is one of the simplest and most common studies which we are designing for the formulation or the tool which the formulation we are developing. Because it helps in observing the groups of people at a single point in time. So it is very common. It is very important to understand that how common a condition is, what factors are associated with it, and that's why we have going for this cross-sectional study, which will take the snapshot of the population at one moment. Okay, that is what the cross-sectional study does. Now you can actually have different types of studies. We will be discussing in second week and third week all these studies, but in the project you will be utilizing the cross-sectional study. So you got the oral cancer chemo prevention. Then you got, now here I have put curcumin, but in your case you will be designing We will take the same molecules, that is the uh, putting uracil acid and EGCG oral gel, where you can see the prompts which are already there, but they are for systematic review and meta-analysis. So if I select the cross-sectional observational study and I ask them to generate the prompts, it actually changed now for a cross-sectional or observational study in oral cancer chemoprevention using the molecules for oral gel. Define the study aim, target population, describe the clinical community setting, inclusion exclusion criteria, sampling, and the recruitment pathway and the single time point. So that is the information you will get from the first prompt. So you will be again utilizing this prompt. You will go into the perplexity. You will add, and again you will go for the deep research, and you will run the prompt, and you will get the information for that study which you are designing with the molecule synthesis. Now, previous session, previous demo, that you should go for deep research, but we have got 100 prompts, right? So should I run 100 prompt one by one? That will be the question. But you can see with the one prompt, you have already got like 10 to 15% of the information which you need to design the cross-sectional study. But the way we have designed this tool is that you don't have to run one by one. There are total 100 prompts which will completely cover the cross-sectional study. But you need to, you can see there is an option here. There is an option here to export the text. Once you export the text, you will have the 100 prompts, and those 100 prompts are divided into category of the clinical protocol, that is study design. So 1 to 11 prompts are for study design, 12 to 22 are for data collection, 23 to 34 I will share this, but 23 to 34 is to understand the bias and the risk, 35 to 45 are for ethical and regulatory, 46 to 56 is to understand the statistical data and what analysis need to perform, and about 57 to remaining are for the implementation in the real world with the advanced layer. Because you need to understand the cross-sectional study is more about understanding the data at one specific point. So we will be designing that for one specific point, and then we will be uh, putting that on the clinical trial website. So that's how the tools is designed. So now I told you that 1 to 11 prompts are for study design. So if you go to the second prompt, so this is the second prompt. Till here it is taking the repeating the first prompt, and then it is asking to turn the previous concept into a SOP style procedure with step by step actions, responsible role, minimum documentation required, and the acceleration triggers. So that will be the second, which will connect to the information that you got in the first. Then in the third one, again, if you see for third one, you the first prompt which we got is still there in here, and then it is asking to define. So first prompt is very important because it is actually giving you a lot of information. Then the remaining prompt are created for making sure that it will connect the content which is there for the research study. Now I will show you one more thing. Then the people who are doing AI in clinical research, they have to also register. I will share that website as well. Okay, so there is a website where you need to upload that protocol, which will be, we will have designed that. Okay, can you see the website? It is loading. Okay, so this is the website which is uh, be useful to actually have the e-protocol writing. So it is a tool created by National Institute of Health, which is an institute in US. You need to sign up here, and then you will be signing in. So once you sign in, so I will create, uh, you know, the create new protocol here, and I will allocate you the protocols. In the meantime, you will already have this information which you will get from the prompts. Okay. So once you create, you are going to connect with me, like these people, team from my, they have connected with me. I will add your new contact once you create your email, the account here. I will create a new protocol and I allocate that protocol to you. So, for example, here, case control observational study. So, this case control observational study uh, help to actually, the information you get with the AI clinical tool help to actually add all the information which is needed in the clinical trial. So, all this information is filled from there. So once you create your account, I will allocate you. Here is manage team. So I will, give the team member as an author, and the sections. In your case, because it is a complete protocol, so all sections will be given to you, and you will be added as an author, and there will be another person who will be a reviewer for the content which you have written, and all the sections will be given. That is the second, and the third will be me, who will be looking at this, and I can directly submit the once I done the complete review, directly submit to the clinicaltrial.gov website. So this is how it will end up the AI um clinical trial internship project, where we will have the protocol design. Simultaneously, you will have your reports and a drafted, you know, uh, article as well, which we can actually publish. Now, this is still only the half done, where you have designed the protocol and keep it ready for the clinical studies. It will continue, and then the data will be generated, which will be then uploaded on the clinical trial website. So this is how the AI clinical trial tool and the project will work. So if you have any question regarding that, you can put in the comment box, and I will answer it. So we have already shared to you the molecule names, which will be in the oral gel. for oral cancer prevention. And I have given you the demo for that one. Okay, so that's the second uh, second tool and the project-based internship demo you will be utilizing in your project. Uh, Now, the third one, which is the AI in pharmacovigilance studies. So the projects are located. I will just open the tool. We have already shared the link to you. Um, okay, so this is the uh, third tool which covers the pharmacovigilance part to identify any adverse effect or any toxicity or anything if there is there with the molecules which we are studying in the disease. So when you start with the um, um, pharmacovigilance from studio. You have got eight sections. Those are the eight modules, which covers the complete report of the pharmacovigilance for that category of the drug. Or if you're developing something new, you can utilize that for that purpose also. So we are developing combinations of small molecules from plant. So we need to understand that will it produce any toxicity. So let's say in our case we are looking for oral chemo prevention. So it might create anything with the allopathic drug, which will be used when you are designing the information for pharmacovigilance. So there are eight categories. I will show you the first part, but then it works on the similar information and it create the scientific prompt to acquire the information from the large language models. In this case also we will use perplexity, but for pharmacovigilance it is good to use Grok as it also connect to the social media that is X. So yes, so data coding is there, signal detection is there, causality is there, risk and regulatory is there, predictive QA and audit is there. So when you are so for the first case and source, I will go with the same um, drugs. That is, I am creating curcumin. Sorry, this is for disease. So I will go for herbs or nutraceutical or the combination. So I'm going for curcumin, uric acid, and AGCG for oral gel. Remember to add oral gel here. Now with allopathic drug, uh, those who are doing pharmacovigilance uh, project, you can add here, but if you don't add, it is still fine. But I will suggest that definitely fluorouracil for oral cancer. So if we change the disease, the cancer category or diabetes or anything other, we will have the allopathic drug because the purpose here is not only to identify the adverse effect of the combination, but simultaneously because we are working on small molecules from plant, does it actually produce any interaction or any issue with the allopathic drug which is given in the combination, and then it will be oral cancer chemo prevention. So you can see the first prompt here is retrieve real world pharmacovigilance case report involving the herbal product and the drug in the disease from the databases and also the social media. Include patient demographic, then E types and the outcome. Now I have not yet generated. So this is the prompt which is there. Once you just need to see here herbal product. If I generate prompt, now you can read it. Retrieve real world pharmacovigilance case report involving the molecules which we have given with oral gel and the drug in oral cancer chemoprevention from the databases. So you need to understand that why it is important because the way you are putting the information is going to give you the data. Now, if I change here, or if I don't, let's say we have not taken any allopathic drug, it will generate the prompt. See, it has given curcumin, uracic acid, and ECGC oral gel, and the drug in the oral cancer. It will now look for random information for any specific. So if you are not working with any standard drug, then you don't have to add the allopathic drug. But if you are already in the stage, so in this case we are already in the stage with the human work, where oral cancer drug which is given to the patient is 5-fluorouracil. So that's why we are putting that 5-fluoro to understand if there is any adverse effect or anything available in the public domain. And then you can copy the first prompt and perplexity. Here I am perplexity you can use for all the areas, but pharmacovigilance, if you have grok also, you can utilize grok for the studies. Prompt is based, make sure you run this. Not only the grok and perplexity, but also the deep seek, which is one of the large language model from China, actually works very good with the PV area. So you should always look for that as well. Now there was a question in previous one of the session which we talking about the theory, that one person asked me that how to decide on the model. So this is how you are going to decide on the model, because it varies based on the areas which you are working on, from basic research to clinical, even marketing and patenting. You need to work out not just the model, but what you give as input. So it is more about the input and the model. Uh, Even ChatGPT can work if you actually have a prompt and have a, information given to the LLM. Based on that, it can give you the content which is relevant. Now, connecting to the literature and connecting to the web search. All model got that now, but with the PV, with the clinical, perplexity, grok, deepseek, cloud, these are very good models. Okay, so you can see here that it has given I could not verify real world post marketing case reports for this molecule in oral gel. If I remove, let's do that actually. uh, what happened we'll see. So instead of, okay, instead of changing in the thing, we will change here only. So I am putting the same prompt, but I will remove all these other drugs with oral gel and also the 5 fluoro, and remove everything and just look curcumin in oral cancer chemoprevention. So this is the information given for the combination with 5-fluoro. You will also see what was found. It has given that it has found something with the curcumin, and we will check that separately as well now. Also, there is a mouthwash with EGCG, so that is there. So that information is there. Practical takeaway it has given, the best strategy it has given. But let's just run without the combination and the oral gel and see what real world pharmacovigilance case report it can give us only for curcumin. Okay, so in this public and the other databases, that is PubMed or VGBase or the social media, it has not found much about the curcumin with the oral cancer, but it has given information which is available or some studies which are available with curcumin and the clinical work which is going on. Simultaneously, it has also given the information from VGBase if there is anything in the case safety report, and it has also gone through publicable accessible sources from 2020 to understand if there is anything with the curriculum. So this is how the large language model work. Again, I will say if you go for deep research, if you go for numbers, so in the sessions I have told you that when you are running the prompt, so in the demo I have showed you with perplexity, but you shouldn't just use perplexity. You should always use minimum two LLMs. So if you are working with predictive, I will suggest that go with Perplexity and Gemini. When you are working with clinical and the prompts, you should go with Perplexity and Cloud. When you are going with the pharmacovigilance, you should go with Perplexity and Grok or DeepSeek. So these are the models which you should always use, and based on that you will get the data. Now that data you shouldn't just copy paste, but utilize it and connect it with your project and the information which you got in the literature mining, and connect the meaningful hypothesis for your conclusion. So that's about the tools, the demo which we have uh, connected for the people who are doing the project which are related to them. Now, if your area of research is different, once we now the tool is with you while doing this project, you can always you know play around with the tool for your project or for your formulation. And anything you observe, you can always connect with us, and we will take it further. So if you have any question for the people who are registered for this internship program from AI, predictive medicine, clinical research, or pharmacovigilance, you can put in the chat box. You can email us, or you can WhatsApp on the numbers, so that way we can answer your queries if you have any. Okay, so let me know if you have any question. Okay, thank you for joining. I will see you on Monday. Monday timings afternoon. So predictive medicine is at 1:30. Pharmacovigilance clinical is at 2:30, and PV is at 3:30. The links are there in the graphy. We will not, we might not have YouTube link. You will be only on the graphy. We will decide that, but at the moment we will be only on the Swalife Academy platform. for the second, third, and the, maybe fourth week as well. Fourth week we might actually have YouTube as well. So thank you for joining today, and I will see you on Monday. Thank you.